Hello everyone. Today's topic is mechanism of stomatal transpiration by starch-sugar interconversion theory. What is transpiration? The loss of water in the form of vapors from the living tissues of aerial parts of the plant is termed as transpiration. Although plants absorb large amount of water from the soil, only a small amount of it is utilized by the plant, and nearly 95% of absorbed water goes out through transpiration. There are three types of transpiration. They are stomatal transpiration, cuticular transpiration, and lenticular transpiration. Among these, stomatal transpiration accounts around 80 to 90% of total water loss from the plant. The loss of water from the stomata in the form of water vapors is called as stomatal transpiration. Transpiration occurs when stomata opens, and when it is closed, transpiration stops. Several theories were proposed from different physiologists to explain the mechanism of opening and closing of stomata. One of such theories is starch-sugar interconversion theory, and it is also known as starch-glucose 1-phosphate interconversion hypothesis. This starch-glucose 1-phosphate interconversion hypothesis was proposed by Lloyd in the year 1908, and according to this theory, the stomatal march, that is opening and closing of stomata, it is due to the conversion of starch into glucose 1-phosphate during light in the guard cells of stomata, whereas its closure is due to the conversion of glucose 1-phosphate into starch, that is during dark. It is based on the activity of the enzyme phosphorylase in the guard cell. During the daytime, in the presence of light, the pH of guard cell it is around 7. During a light condition, the insoluble starch it will be converted into soluble glucose 1-phosphate, and this glucose 1-phosphate dissolved in the guard cells, and this results in the increase in osmotic concentration of guard cell, so that the osmotic pressure, and it decreases the water potential of the guard cell. Consequently, the water moves into the guard cell from the surrounding mesophyll and epidermal cell, so that the guard cell it will become turgid and the stomata opens, so that the transpiration will takes place. Whereas during night condition, that is during dark, the pH of guard cell falls down to around 5. At this condition, the enzyme it converts glucose 1-phosphate into starch. Here, as a result of this, the water potential of the guard cell it increases, and the water moves out of the guard cell, and the guard cell it will become flaccid, so that the inner wall of the guard cell it come close to each other, so that the stomata closes, whereas the transpiration stops here. The concentration or the pH of the guard cell it is controlled by the concentration of carbon dioxide. During daytime, as photosynthesis takes place, carbon dioxide will be consumed, so that the less amount of carbon dioxide it is available for the carbonic acid formation. So, the guard cell it will become turgid and the stomata opens, and here the one more condition, that is the starch will be converted into glucose 1-phosphate and the transpiration takes place here. And the low pH is due to the conversion of glucose 1-phosphate into starch, that is during daytime. Here, as there is no photosynthesis, the more amount of carbon dioxide it is available here for the carbonic acid formation. So, the stomata close here and no transpiration. Now, let us compare the two conditions, that is guard cell in light and guard cell in dark. Here, during a daytime, that is in the presence of light, respiratory carbon dioxide is consumed in photosynthesis, whereas during dark, respiratory carbon dioxide accumulates in the intercellular spaces. During light, the pH of guard cell rises, whereas during dark, the pH of guard cells falls. In the presence of light, acidity is decreased and so starch is hydrolyzed into sugar, whereas during dark condition, the acidity is increased, so sugar converted into starch. As the osmotic concentration of this guard cell is more, water diffuses into the guard cell from the nearby mesophyll cells, so the turgor pressure and volume of guard cell increases. Whereas in dark condition, the water diffuses from the guard cells from nearby mesophyll cells, so the turgor pressure and volume of the guard cell decreases. Inner wall of guard cell move apart and stomata becomes open during light condition, whereas during dark condition, the inner wall of guard cells come nearer and stomata closed. During light condition, as stomata opens, transpiration begins to take place, whereas during dark condition or during night, transpiration begins to cease. This is all about the mechanism of stomatal transpiration by starch-sugar interconversion theory. Thank you.